Good morning. My name is Laura Cottino, and today I will be presenting my work entitled An Assessment of the CNV Landscape in Sub-Saharan African Populations using Short and Long-Read Sequencing. Various types of genetic variation exist within the human genome, and while single nucleotide variation has been extensively studied, an assessment of the CNV landscape is lacking, particularly in African populations. Additionally, most studies on CNVs have been done using genotyping arrays or short-read sequencing, which pose major limitations to CNV calling. A solution to this is the use of long-read sequencing, which allows for far more accurate and sensitive CNV calling. Therefore, the aim of this study is to assess the CNV landscape in sub-Saharan African populations using both short and long-read sequencing approaches. Moving on to the methods and materials. 919 high-coverage whole genome sequences from individuals across SSA were used, and CNVs were called using genome strip. PacBio smart sequencing was performed on 8 of the above individuals, and structural variants were called using PBSV. This slide shows the results of the short-read sequencing data. A total of 15,658 CNVs were identified, the majority of which were deletions. Duplications were found to be significantly larger than deletions, and just under 30% of the CNVs were classified as common. When looking at the size distribution of the short-read CNVs, you can see that the tool used is not capable of detecting small CNVs. Figure 2 shows the most severe consequence of each CNV, and 9% of the CNVs affected the coding region of a gene. Lastly, 4.8% of the CNVs were classified as novel. Moving on to the results of the long-read sequencing. Just over 42,000 CNVs were identified from only 8 sequences, and the number of CNVs detected per sample, as well as percentage of novel CNVs detected, were also much higher when compared to the short-read sequencing. If we look at the size distribution of the long-read CNVs, you can see that just under 92% of the CNVs were less than 1,000 base pairs in size. Lastly, when comparing the short and long-read data from the same 8 individuals, just under 50% of the short-read CNVs were concordant with the long-read data, but only 3.9% of the long-read CNVs were observed in the short-read data, and the greatest gains in sensitivity from the long-read approach were among small CNVs. CNVs are an important yet understudied source of variation, particularly in African populations. We identified a large number of CNVs from the short-read sequencing data. However, long-read sequencing was able to detect far more CNVs from far fewer samples. Currently, our understanding of the full spectrum of structural variation is incomplete, and long-read sequencing of additional samples, particularly of African ancestry, will further improve our knowledge. Thank you.